Hello friends, welcome to BMH Learning. This video is regarding X and V factor test. X factor and V factor test is done for the identification and differentiation of Haemophilus species, especially identification of Haemophilus influenzae. H. influenzae is a fastidious organism that requires both X factor and V factor for growth. Basically, a lone of the test organism is streaked onto one of these agar mediums. The discs impregnated with XV or both XV factors are placed directly on the confluent inoculation, allowing diffusion of the accessory growth factor into the medium. The organisms will grow only around the disc that provides the appropriate factor for the growth of the organism. That is, the presence of growth around the disc but not elsewhere on the plate indicates a requirement for that particular factor. Procedure: using aseptic technique, select isolated colonies and prepare a suspension equivalent to a 0.5 McFarland standard. Dip a sterile swab into the organism suspension. Inoculate by streaking in 3 different directions by rotating the plate in a 60-degree angle after each streaking. Streaking in this manner ensures confluent growth. Allow the surface to dry for 3 to 5 minutes. Place the X, V and XV factor discs on the agar surface. Incubate overnight at 35 to 37 degrees centigrade. Observe for growth or no growth around the discs. Result interpretation: positive. Growth around the XV disc only shows a requirement for both factors. Growth around the V disc, no growth around the X disc, and a light growth around the XV disc shows a V factor requirement. Negative. Growth over the entire surface of the agar indicates no requirement for either X or V factor. This was all. Thanks for watching.